No, it's working. Yeah. Whatever happened
Boa.
didn't happen. It's just configuring something or whatever. Ricardo, can you test if it's live on YouTube? But it seems to be.
Yes.
Okay. So, I think this means we can start. So I'll start recording here as well, just in case.
Just confirming we are live.
Okay. So, well, hi everybody. I'm Olavo, we're Brisa, and we're live, and thanks for being here, both for the ones speaking and the ones listening and hopefully asking questions later. I'll try to share my screen here, so hopefully you can see my slides now. Is that okay?
Yes.
Okay. So yeah, we're Brisa, and I'll make a very quick introduction of the group and what we do and why we're here, and then pass the word to the real speakers, who are Mariana Abreu from from Brisa itself, Carly Huismans from Hadwood University Medical Center and Circle, formerly Thais Frederic-Gavon from Unicamp, and Alexandra Bennett-Brown from BIH, Quest, and Camarades Berlin. And so we're Brisa, that stands for Brazilian Reproducibility Initiative in Preclinical Systematic Review and Meta-Analysis. It's just a pretty acronym, and Brisa means like a light wind or breeze in Portuguese. We're currently an online collaboration of a little bit more than 40, I think 41 people, mostly from basic biomedical research, who are interested in systematic reviews, who are not like formally systematic reviewers by official training, but most of the people have learned on the way. We started out very informally as a spin-off of the Brazilian Reproducibility Initiative, which is a large replication project which is currently finishing in Brazil, where should be have our results till the middle of the year. But we had over 50 labs within the initiative for most of it. As we're about to start in the pandemic in 2020, everybody closed down their labs, and like we were stuck with like a big network of people and no labs to experiment. And most people were kind of well, they were going through all kinds of troubles during the period, but like most of them have had a lot of time to spare. And at some meeting, at one of these general meetings, like, okay, what we do with this? If nobody can work on the lab, what should we be doing? And somebody, I don't know who actually, so like this is somebody's idea. We have to figure out who proposed that. It's like, yeah, we could do some systematic reviews, but nobody had real training in that. So we tried to get, we figured out who was interested. We got a group of like around 20 people, and then we found people who could actually like help train people. And very informally, we started out on 3 collaborative projects, whose topics were actually like decided by the community through a somewhat democratic way, and that kind of worked. I think people were excited and happy about the process, and we kind of realized that there was a niche to be filled, both in actually training biomedical researchers in systematic review methodologies. This is what we're here for, and in catalyzing collaborative projects involving systematic reviews. Those can easily be done at a distance online, so we can actually provide a network to like get interested people to work within a project which usually requires a lot of people. And perhaps more importantly, we realized that those two goals actually complement each other very well. The best way to train people is actually to get them working on something. So we figured out that we had something going, and it happened spontaneously the first time around. We didn't really plan for this, but we tried to figure out what worked and decided to replicate it in a more structured format for a whole new group of researchers last year. So as the group was dwindling down and some people and the projects were ending, we put up an open call for new members last year. And set up a structured online training program with recorded classes, with live classes, with some supporting material in Portuguese to make life easier for new people joining. This program was specifically focused in systematic reviews within the basic science preclinical context. And we then launched an open call for new members. We got over 100 applications. We selected 30 people who went through the course, which took around 3 months. Mariana will talk more specifically about this. And these people are now engaged in Brisa systematic review projects. We have 6 of them currently ongoing, along with the original members. You can take a look at that on our website. So we have actually two reasons to be here today. The first one is really to reflect on that process, to discuss what worked and what didn't, and to exchange ideas with people who have tried different approaches to train people within systematic review methodologies. So this is like the main focus of the topic today, of the symposium today. But we're also here to actually advertise the fact that the material for the course is now live and accessible online via big YouTube playlist. We have the supporting material on Google Drive as well. It wasn't meant to be a massive online course, so it might have some rough edges, and if you find some rough edges or something that doesn't look right, please email us and tell us. I mean, this is actually just like transposing the material for the live course to a massive online format, but it's there. I mean, it's ready. There's no reason not to share it, and we hope that might be useful for some other people. We don't really plan to have a new live course until 2024, although we probably put up an open call next year. And this is about it. And really, the reason I think this is important goes actually beyond systematic reviews. I think actually training people in this kind of approach actually makes them better researchers. There's some data on that, on the impact of training researchers on systematic reviews and how that actually changes their approach to research and to research reporting. I do think this is actually I mean, it's very limited empirical evidence, but my personal view really kind of goes along with this. I think we can make better science by helping people to be more systematic in the review approaches and to actually have them take a look at the literature and see what's wrong with it, because there's a lot wrong with it. And I think doing a systematic review really allows you to see all the reporting gaps and the problems we have to solve as a community to make more transparent, better research. And just as we're talking about better research, we're also launching the Brazilian Reproducibility Network next week on Wednesday at noon. If you want to come along with this one is in Portuguese. But it's going to be, I mean, it's just meant to be a reproducibility network, joining together different stakeholders, groups, institutions who are interested in promoting research, reproducibility, and transparency. Brisa was one of the members. There's around a dozen other members, and we're actually putting up a call for new members. So if you want to learn more of how to join a bigger reproducibility network that spans a wider array of disciplines and types of projects and initiatives, please join next week at noon via this link here. I'll put in the chat as well. And this is about it from me. So I'll pass the word to Mariana, who present the Brisa course, and we'll then go to the other speakers who present their own experiences. Mari?
Can you hear me?
Yes.
And the presentation is okay?
It's there, but it's not full screen.
Okay.
Now it is.
Now it's okay. So hello, everybody. Well, Olavo has talked overview about how Brisa started and how we keep working during these 3 years. And I'll talk more specifically about the Brisa training program that, as he told, was also a way to get new members to Brisa and to keep Brisa alive. So we started in this context in June 2020, in the context of pandemia and lockdown. And in a brainstorm, we realized that many people in the group, from researchers from Brazilian Reproducibility Initiative, would want to get more experience in systematic reviews. So we decided to take the first steps to make it possible in that group. And since just few of us had experience in systematic reviews and meta-analysis, we decided to invite people with more experience. That's how Alana and Gabriel got into the group. And they realized that if we wanted to start some projects, everybody should have at least basic information concerning systematic reviews and meta-analysis. So they prepared 3 classes in full around the 6-hour course, just to give the basic information. But the idea was to start producing projects together. And so we opened the possibility for all the members of Brisa at that time to propose projects. In that time, we were around 20 members, and we had 8 project proposals. The idea was to develop projects that would take around 1.5 years or 2 years. And we had presentations of these projects, discussion, and then in the end, we decided to start developing 3 projects. 2 of them are still being developed and haven't finished yet. And we are a little bit late. And that's something that in the new projects, we are taking care to try to make projects that are really possible to be finished in 2 years. And the sleep deprivation project, for now, it has been stopped. We are not sure if it's going to be, it's going to return or not. After 1 year that Brisa was going on, we figured out that maybe we need new members because some people were leaving, some people were not with the same possibility of participation that they had in the beginning, especially because the world had changed a little bit. In the beginning, we had a context of lockdown, everybody not going to the labs. And here in 2021, it was kind of different already. And we thought that the way we did the training was interesting. People were really learning by doing the projects and having the help with some people with a little bit extra experience. And so we decided to do it in a more organized way. We took a little longer than we thought to prepare the training program. We took around a year to prepare, especially the recorded classes that took a little longer. And the idea is that the training program would be composed by the online course and the project development with assistance of Brisa, so that the developing the project should be also part of the training. At that time, we were around 30 members in Brisa because some people entered in the beginning of the projects. And 9 of us were directly involved in the preparation of the training. So the online course was composed of recorded and live online classes. We also prepared some proposed assignments that would be given for people. And then in the live online classes, we would discuss and correct these assignments. And the online course was developed from August to November 2022. It took around 2.5 months, more or less. And during this period that people were taking the classes, we also start choosing the projects to be developed. When we were doing the selection of people to participate in Brisa training program, we already in the beginning asked for people to propose projects if they want. And the idea is that this new round of projects would be projects proposed by the new members, but with the involvement of original and new members. So just making a step back here, concerning the selection, people could do inscriptions by Google Forms from May 2022, from May 2022 to July 2022. We asked for name and email, also the curriculum, the current position, and also a letter of interest with a small summary of the reasons why they would like to participate on the Brisa. Previous experience in topics related to meta-analysis, and the project proposal that was optional. we had 124 candidates to become Brisa members. And these candidates, most of them had doctorate degree, more or less half of them, and about 30% had a master degree. Concerning their position at that time, most of them were postgraduate students, and around 30% professors or researchers. We had a selection committee composed of 6 members of the original group of Brisa. And each candidate was evaluated by 2 members of the committee. Sorry. And after all the candidates were evaluated, and we had given grades for each member that was evaluating, give grades for them, we had a mid discussion and to decide to make a final decision. I'm sorry for that. On the 29 new members on Brisa. So concerning training degree and the position they were occupied, more or less we follow the same numbers that we had in the full group. And we also tried to include members from every part of Brazil. So talk a little bit more in detail concerning the different parts of the training program. For the course itself, the recorded classes included basic information on the main topics on meta-analysis and systematic reviews. We did videos from about 15 to 20 minutes. Depending on the topic, we had from 1 to 2 videos for the topic. And the videos were available for 1 or 2 weeks. In the moment that we released the videos, we also released the assignments related to that topic. And concerning all the recorded videos that compose the course, we have around of 6 hours of recorded course for people who are interested in now doing by itself. And in the moment the course was offered, we used the Wix platform to put the videos online. So in the end of each period that for each topic, 1 or 2 weeks, we had live online classes. And in these classes that took around 90 minutes in Zoom meetings, we would discuss the assignment that were proposed, correct it, and have a space for doubts and discussion. All the live online classes were recorded, and they are also available online now. And we have around 16 hours of live online classes recorded. The second part of the training concerned the development of the project. So in the same time that people were doing the course, we had some meetings with new and old members. And in those meetings, 9 project proposals were presented. The person that proposed the project would make a small presentation, putting a little bit more details on the project proposals. And the discussion was based specifically in definitions of strengths and weaknesses of each project, and also the feasibility assessment to try to make projects that could get an end in 2 years. So in the beginning, we had 9 projects. In the first discussions, we have 5 projects being carried forward, and later one more was cut down. So we defined 4 new projects to be developed in Brisa. And all the new members were distributed in these 4 projects. Also, old members are participating in these new projects to keep the idea that people with more experience can teach people with less experience, and also be responsible for the organization of the projects, or at least help new people that are responsible for that organization. The project development part of the training started in January 2023, and is still going on. The project development is based in individual group meetings, and also in asynchronous communications via Zulip. That is something that it's helping a lot for tasks to be proposed and doubts to be solved, and also for people from other groups to be aware of what's happening in the other groups, and also give their opinions in some specific points if they want. We also have month Brisa general meetings. In those meetings, each group usually give an overview of what is being done during this month. And it's a space for also to everybody to put their opinion in all the projects, and also for those projects to be developed in Brisa as a bigger group. So as Olavo has told, now we put all this material in open access. The recorded classes are available in a YouTube playlist, and the assignments and the live online recorders are available in a Google Drive. And well, the idea is that people, if they want, they have this material to make a individual learning until probably 2024, when we are going to open a new training program in Brisa and open space for new members to get into Brisa. So that was our experience. Thank you very much for your attention. I just open here the YouTube and for you to take a look. So here all the classes are put in the playlist. We have put in the divulgation of this workshop here the link for that playlist, but we can put again now in the chat, and also the link for the material that is organized here, including an introduction, and then each topic with the classes that are in YouTube or are also here in the Drive, and the material from the online live classes recording, and the material that are needed to follow those classes. So thank you very much.
Thanks, Mari, and welcome everybody who has arrived in the middle of the talk. We're continuing now with Caroline Huysmans, who talk about her own experiences in training people in systematic reviews, which is quite wide, and we're very much looking forward to hear you. I think you're mute, Caroline. Can you unmute yourself? No, I'll do it. Wait. Let me. I figure out the co-host could do this, but I'll just try now. You're still mute.
Also try to do that.
Yeah. Thais, can you unmute yourself? Just to see if it's her own problem or...
Yes.
Yeah. That's weird. Should we change the order while you try to log in again, Carlin?
Olavo, maybe let's try to
I've done it already.
put that everybody can unmute themselves.
Everybody can. So I'm not sure what happened. Maybe it's the other Carlin that's... Can you try to log in again, Carlin? I think that's the safest option to... Do you hear us? Still mute. Yeah. Try logging out and in, and we go on with Thais meanwhile. Okay. Sorry for... Yeah, I don't know what happened. So... Thais, thanks a lot. You're mute, but I think I guess you can. Yeah, great.
Can you hear, see?
Yeah, we can.
So thank you all for the invitation and also for our audience. So my name is Thais. I'm a professor at the Faculty of Pharmaceutical Sciences at University of Campinas in São Paulo, and I'll try to talk about what Olavo asked me. So how to best train researchers in systematic review based on my experience, which is not on preclinical studies. So it's in systematic reviews, basically epidemiology, human studies. The first method that I would recommend, and it's a bit of conflicts of interest, if you may see, so it's to do our course, systematic review and meta-analysis. It is in Portuguese, but it has subtitles. Some interviews are from researchers and are also in English, but we have audience from other countries, and it's very well approved, and we have a lot of good feedback from this course. So this would be my first suggestion of how to train to do this free online course, which is available at Coursera. And we have an introductory course. It's health-based evidence, and it gives main methodological concepts about evidence-based medicine. So about our problem, the problem that we have, so we should have better researchers, better systematic reviewers, so that we can have good quality systematic review. This is a very wide known research about the mass production of reductant, misleading, and conflicted systematic reviews and meta-analysis, which was the author is Professor Ioannidis, which is an authority in meta-science, meta-research. And the conclusion of the paper is that we have many systematic reviews. So we have a grown number of systematic reviews, as you can see in this graphic, from a search in PubMed from 1986 to 2014. So we can see we have the growth is really huge. And about the quality, the researcher selected systematic reviews about genetics medicine, and he classified, he did a grading using this category. So it's somewhat subjective. Everything in this area, even systematic review, we can say they are transparent, reproducible, but it's done by human. So as done by human, it can have flaws, can have subjective assessment that can change also the methods, judging in different ways, but based on his experience and his classification, he pointed out that in this area here, it would have about 3% of decent and clinical useful systematic review, so that which are not redundant, that really add knowledge based on what is already known. So there is a lot of redundant and unnecessary, according to his scale, it's about 30%, has flaws that cannot be repaired or not published. So a lot, as you also mentioned in your first experience in Brisa. So starting a project is something that we can do and we have a lot of energy, but to conclude it is sometimes it's not very easy. So 20% is not published, and also others, 13% are abandoned, are misleading due to abandoned genetics. And so we surely have a problem in the product, in the systematic reviews based on this assessment. Also, we have other literature that shows this, but this, I think it's very, very interesting for us to start this. When I talk about systematic reviews, I usually say that sometimes it can be a letter soup. So a soup full of letters, basically, because we do have a lot of instruments, a lot of tools, many of them validated, that really helps researchers to follow the methods, to properly communicate. But sometimes we have, it's like we have a use of these terms or methods that are not reasonable for that systematic review. So your systematic review is for a prevalence study. So you should not, for example, search first for randomized clinical trials that do not have representative samples, are based in convenience samples, and so on. So sometimes you can just repeat and employ these methods, but it's done uncritical. And so that would explain this image of a soup of letters. This is a first a main diagram that me and my colleagues, when we were elaborating our course, we thought how to just in one image to bring the main steps of a systematic review, and the size of the circles would represent the demand of work, so the workload in each step. So the first one, one of the biggest, is on the PICO question. So in PICO question, we are basically saying not just to elaborate the PICO question, the population, the intervention, the comparison, or the outcome, but mainly to plan the research to see if there are studies for you, because sometimes what really is needed is not a review, because nobody has researched this. So how can you review something that's not available in the literature? So it's checking if there are studies about this, if it's feasible, if you have the structure for it, to also to do pilotings of the search, checking if your planning is really feasible. So this, and also starting the protocol, not yet registering, because sometimes people have an idea and the idea is very good, but so much when you start piloting or doing the first steps, you see you have to correct it, and then it's recommended that you piloting, and then when you are more aware of your conditions and of the field, then you would register this protocol. But this moment of planning that is here available in this first circle is where we consider that we need a huge workload. So we should spend a lot of time in this moment, because it will drive all the steps following that. Of course, you can, it's an iterative process. You can return and correct and so on. But if you spend a lot of time here, the chances that you can anticipate problems, it's more, the probability are higher, so that you can anticipate and then avoid your project to be finished or have problems in the future. And then we have searching the literature. It's a considerable amount of work, but it's not that much. So based on your PICO question and with the help of a librarian, someone experienced on that, and also starting doing a pilot search and then peer review the search, following, for example, the press guideline that someone outside your group would review it and check for filling in a checklist to check if your first search done in just one database is enough to identify the main studies. So how can you know which are the main studies? So this always starts in this first step. So you have, like we call, sentinel studies, which are studies that are like indicators that your search is well done. So when you search those studies, if they are indexed in this database, your search should identify this, should retrieve these studies. And then, so it's, of course, it's a critical step, but it's not too much work. So it can be done with relatively small team, experience, and then check and then expand these results to other databases after you really check the validity of your first strategy. And then there is the selection, which the gold standard is done by at least 2 independent researchers that would check the eligibility of their studies based on your, while you decided on your project, again, in the first circle here, the PICO question. Then the data extraction, which has a lot of workload. Sometimes we have to contact authors, you have to do conversions, have to do transformations, admit some standardizing in your work. And recommended is done also by 2 researchers independently. So it's a lot of work and it's duplicated. And then there is the risk of bias, so that you can judge the quality, the methodological quality of the included studies using validated tools. And then the synthesis that mainly is known in the meta-analysis, but also sometimes the tabulation of the results in a standardized way so that the reader can understand the result in each of the included studies. So sometimes we can see a table or another way of synthesis, but the metrics is not the same. So the reader, the person that will use the systematic review, gets really confused. And also in this step, the meta-analysis that would require good data extraction in this in the former step. Then in human studies, studies that do recommendations, recommend the use of a technique of a medicine or a prevention prophylaxis, we should assess the certainty of evidence. So we done we have a new result, and then we have to inform the reader about our certainty on the evidence based on the risk of bias, but also in the heterogeneity, in precision, and other factors that would affect our confidence in the results. And then the scientific writing, which is a common step for any research, but it's also very time consuming and requires that the team properly reports clearly, concisely. And here would use one of those tools that's very well known, the Prisma reporting guidance. So here it would be, here is the step that Prisma would be used. Sometimes we see systematic reviews that says, oh, our methods are based on Prisma, but Prisma is a reporting guidance. So this is an example of how this using of jargon, sometimes uncritical, can be misleading. And then publishing, which is also very much time consuming. Systematic reviews sometimes are not, some journal may not consider as original research, or other charge a lot for publishing. And so it's sometimes you should consider, if it takes too much time, consider updating the search, keeping regular updates so that you can incorporate new results. So in a nutshell, showing that a lot of knowledge and a lot of the logistics that would require at each step for doing a good quality systematic reviews. So what I would recommend, or what I would say that are methodological skills for systematic reviewers. So basically, this research should have good good skills on epidemiology and research methods. So it's epidemiology understanding, the study designs, that the main study designs that are used for assessing data, so observational studies, cohort studies, randomized clinical trials, which are the main biases, main limitations that would affect the design of these primary studies, so that this person can design the steps, but also to assess how this evidence that it's being assessed is biased or not. So it's basically reasoning about research methods, and the scientific reasoning about in systematic review. So understanding the question framework, understanding if I want to know if a technique is a diagnosis technique is better than another. Sometimes it's not a randomized clinical trial that would give me this evidence. Sometimes it's a cross-sectional study that had done this assessment of two tests, gold standard and index test. So sometimes you have to develop this scientific reasoning and check if your question frameworks, which is the best design, which one is the best source of data. So should I search in Cochrane or no? I wouldn't have clinical trials or systematic review. It's not interesting. So I do not spend a lot of time and energy in something that's not is not related to my question. And understand also the type of data that I should extract from studies. So based on this reasoning and the epidemiology and research method skills, understand which data would be collected and how it would be appraised, so that it brings the required information for the results.
Thais, you're already over time, so if you can just move quickly and.
Oh, sure, I'm finishing. And other reporting skills for systematic reviews, as I mentioned, scientific writing, so the clear and objective writing, adherence to the reporting guideline, as I mentioned, the Prisma, and for qualitative systematic reviews. And if you want to know about other writing guidelines, the Equator Network is the best source for it, so for primary studies too. And the main, besides knowing this step and having this, have a good support for the researcher. As Olavo mentioned, a good way to train research is doing this in practice. So during the research planning, follow the FINER framework. So choosing feasible, interesting, novel, ethical, and relevant research questions, and having the support of an experienced group or a supervisor that can provide frequent feedback, is available for doubts, and is available also for the challenges and to correct again, so that this process is more fluid. And also is not based on the skills that the research should have, but the research support is very relevant. Sometimes people would say a systematic review is done just by the person, the computer. Okay, but we have to give good quality resources so that the researcher can do his job in a proper way. So we have to provide the structure, financial, human training and time so that these resources are available, and some basic, for example, in the material resources. So we should have good quality computer equipment, softwares, cloud storage, also access to printing, translation, and eventual services to give support for the researcher to do his job. So this is what I think that's the best way to train research and systematic reviews, and I'm available for any doubts in the end.
Thanks a lot, Thais. Sorry, I didn't introduce you because of the confusion, but Thais is a professor at Unicamp Pharmacy, and she has actually, I think, the best and most watched course in systematic reviews in Brazil, although I think it's on the clinical side, but definitely worth checking out. So, Carlin, are you?
Yes, I think it's now working. Yes.
So now
Great.
we go. I'll be sending out like 5 minute, 3 minute, 1 minute warning for time. I didn't. Yeah.
Yes, that's fine. I'll start sharing. Just check if it's working. Do you see my screen?
It's not full screen yet, but
I'm trying. Is it full screen?
It is.
Yes.
Okay, that's great. Now, first of all, thank you for introducing me and inviting me. And it's really a pleasure to be here today and tell you a bit more about our experiences so far. But who am I and where am I coming from? Now, I'm Carlijn Hoijmans. I'm from the Meta Research team in the Netherlands. And you see here a photo of me and my colleague Kim Wever. And together we run the Meta Research team. And previously, we've been working for CIRCLE, the Systematic Review Center for Laboratory Animal Experimentation, which was founded in 2008. But a few years ago, we were dissolved, and then the two of us started a Meta Research team. And we are, Kim is a biologist by training, I'm a biomedical scientist by training, and we conducted for many years many animal experiments ourselves. And already during my PhD, I was inspired about the value of the existing literature and the challenge of really investigating this and obtain reliable overviews. At that time, I was not aware of the systematic review techniques, but I tried to do my best with narrative reviews. But after that, I heard from my promoter at the time about the systematic review field. And it was therefore not so much surprising that I started my first postdoc at the time in this field. And over the years, we've been focusing on developing methodology for the conduct of systematic reviews of animal studies specific. And we developed workshops, e-learning, and many other educational programs, of which I will tell you more in the next few slides. And quite recently, we became also the Camerades National Coordinating Center in the Netherlands, of which we are, of course, also very proud. But I would like to start with some history about our team and what we did and what others did before we could really start. And in the beginning of the 70s, probably most of you know by now that Archie Cochrane published a review in which he highlighted the ignorance of evidence in healthcare. And Archie Cochrane, criticized the medical field mainly for not organizing a good system for regularly, critically assessing all the available evidence. And from that moment on, things started to change. And in 1992, the first Cochrane Center was founded. It took almost 10 years before the first systematic reviews of animal studies was published. And after that, this field of animal SR started to evolve. Camerades was founded around 2004, and they initially started with systematic reviews of animal studies in the stroke field. And as I mentioned, CIRCLE started in 2008, and we focused at the time mainly on education and tool development. And if I'm looking back on what we did at CIRCLE and nowadays the Meta Research team, the main milestones for us in our team were political. So at the time, we started to create awareness regarding the need of systematic reviews of animal studies. And the head of our department at the time, Merel Ritskes, she invited members from the parliament to our animal facility. And following this visit and in collaboration with the Animal Rights Party in the Netherlands, 2 motions were submitted to Parliament and both accepted. And in this first motion in 2012, adopted in 2012, I have to say, the government was requested to make systematic reviews mandatory for animal experiments, as had already been the case for many years in clinical studies in the Netherlands. And the second motion was submitted in 2014 and accomplished that systematic reviews were made a compulsory subject in laboratory animal science courses. And what is nice to know is that the laboratory animal science courses are compulsory for everybody who's working with laboratory animals in the Netherlands. So as I mentioned, these motions, besides awareness, also created funding possibilities for us, which could be used subsequently to further develop the field of preclinical systematic reviews. And since then, we as a team started to develop tools for systematic reviews of animal studies. We started to support and coach other people when conducting their SRs, and we developed a lot of educational programs on systematic reviews of animal studies. Now, regarding the tools, briefly, a few tools from our team. We didn't really start with the starting guide, but we wrote a starting guide, which really summarizes the basic steps of systematic reviews of animal studies. We developed a protocol for systematic reviews of animal studies, which can now also be published in PostPro. Step-by-step guides for searching, search filters to identify all animal studies, because as most of you probably know, just by checking the box, finding all animal studies in PubMed, you won't retrieve those studies. We developed a risk of bias tool for animal studies, for intervention studies specifically. We started with developing guidance on meta-analysis and the first steps for grading. So this is interpretation of the systematic review results. As you heard Thais talking about, it's not only risk of bias, it's also precision and indirectness and all these factors that need to be taken into account. And how do you do that? So these initial steps on how to do that for animal studies have been taken, and multiple more tools have been developed. But as mentioned, we also support and coach scientists during the conduct of the review, and we develop many educational programs. And with regard to the education, with these grants I just mentioned at that time from our Ministry of Economic Affairs, because it's a little bit strange, but that was the ministry responsible at the time for enforcing the Animals Experimentation Act. And we developed an e-learning module on preclinical systematic reviews. And it was launched in 2015. And this became a compulsory component of all laboratory animal science courses in the Netherlands. So that was a nice start for us. But we have many more educational programs, and over the years we started to work with many organizations worldwide, and this is still continuing. So now I wanted to go a little bit more in depth in the different kind of educational programs we have. They are not all here, because when I was listening to the others, I thought, oh yeah, this we do as well, and this and this and this. But a few that I wanted to highlight are mentioned here. So we have these, besides the e-learning, many different types of workshops. So we have a general workshop on which I will talk a little bit more in detail after this slide, which takes 1 day or 2 half days. And we have many workshops which are more focused on specific steps of a systematic review. For example, a 2-day program on searching, together with EFSA and many other international collaborators, also a few of them like Alex present here. Workshops on critical appraisal of primary studies, meta-analysis, etc. But we've also focused both in our Masters of Medicine and Biomedical Science on principles of research rigor and robustness. And as mentioned, a lot of coaching of the conduct of systematic reviews. So here you can see the program of our basic introduction course. And as you can see, it's a mixture of lectures and hands-on work. And sometimes we work in groups, sometimes online. But we experienced that especially the variation in lectures and practical hands-on work keep the participants involved. And this was, of course, during the COVID problems, very important to really keep them involved, and especially make them to keep their cameras on so that we really know they're still there. Lectures and practical assignments mainly focused in this course on the introduction, searching, data extraction, risk of bias, and meta-analysis. And here you see the second part of our course, and this is just the general introduction course. And this course is part of a larger program and belongs to one of the funding schemes from our ministry. So it's called More Knowledge with Fewer Animals. In Dutch, it's called MKMD. And since 2014, this call for funding focuses on the effective use of results from animal studies by increasing their retrievability and reusability. And this is, of course, exactly what we try to do with the conduct of systematic reviews of animal studies. So this grant scheme encourages researchers to attend a workshop, the workshop I just showed you, and they also receive funding subsequently after finishing those courses to conduct a systematic review. And when they receive this funding, they need to include in their team experts in the field. And ZonMW is paying for both the course, that is our funding agency, for both the course and the coaching and the teaching, and also the supervision and the coaching is generally done in the Netherlands by either me or Kim, my colleague, so together. Now, in 2020, the impact of this call, which started in 2014, have been evaluated by Julia Minon, and we just saw by Elavo one of the papers in which she summarizes the main things from this report. And our courses and this program have been evaluated, and in the next few slides, I will tell you what the researchers mainly learned, what I learned, etc. So first, what did the researchers mainly say that they learned? And they explicitly mention that their awareness of their field increased. And they thought they were already, most of them experts, but still that increased a lot after conducting these systematic reviews. They obtained more insight in their knowledge gaps, and they become also much more aware than they previously were in the heterogeneity, and especially the between study heterogeneity. They also noticed that finding all animal studies is not easy, and that everything that they learned before during school or during practical assignments themselves, that creating a comprehensive search is really challenging and costs much more time than they expected. And most of them, and especially most of my students, they are all disappointed after conducting a systematic review because they thought that the quality of the literature, of the animal literature, was much better than they experience when they finally start assessing the risk of bias. So they concluded that reporting guidelines are essential, and the research quality can largely be improved. And finally, they mentioned that the animal models used in the studies they analyzed often do not represent the human condition. So really large issues with the external validity. They mentioned, for example, that the induction of the disease does often not model the human situation adequately, or healthy instead of comorbid animals were used, hampering external validity. And they become much more aware of all these issues, not only for systematic reviews, but also when they started to conduct their own systematic reviews later on. And how did it impact their research? Now, they started, most of them, because everything what I'm explaining now is what the majority of the people said, of course, who were interviewed. It's that they started to use reporting guidelines. They improved really their own study quality. They started to implement better methods for randomization and blinding. They became more aware of the various models, and decided which models are really appropriate and which are not. And finally, they mentioned that the awareness of the current situation in their field, as mentioned, really helped them to decide how their future research should look like. They also mentioned that they became more critical. They developed a critical attitude regarding the internal validity, external validity, and reporting quality. Which in turn, they mentioned themselves, improved their capabilities when reviewing manuscripts. So not only, again, in the systematic review field, but as a scientist in general, they obtained many new skills. They also mentioned which skills they really learned during the courses, and the learned and improved skills, and we have some completely new skills. So the new skills are the lower ones. They were mainly about how to extract data from a publication, how to deal with the various data out there, the different kind of software. Most of them are aware of subgroup of meta-analysis, but not how to deal with subgroup analysis and the variation in the statistical analysis you can use. They also learned a lot about phrasing an adequate research question. And what I still find very interesting is that this, and Thais also showed that already, should receive much more attention than it currently does in research. And it's one of the most important steps when you start any research. It doesn't really matter, but definitely also a systematic review. And structuring results, really systematically analyzing your results, was for most of them quite new. It was also evaluated for which topics the participants experienced the coaching to be most useful, because, as I mentioned, we coach all these Dutch scientists who want to conduct their SR, because SonMW, our funding agency, says it's okay if you conduct an animal systematic review, but we want to have that these scientists have, in a way, a kind of stamp regarding the quality of their systematic review, and the methods need to be okay. And they mentioned that they mainly learned or needed the coaching for the protocol development and developing the comprehensive search strategy. The risk of bias assessments is often quite challenging, because most people thought it was just a kind of reporting quality exercise where it's really a judgment and you really need to learn how to do that. And of course, they needed the coaching mainly for the meta-analysis. And this is actually in line with my own experiences, as I find coaching during the protocol development and creating the search strategy the most time-consuming parts over the years. So what did I personally learn?
Also, so maybe this is the last line, but yeah, your time's up. So if you can.
Yeah, I'm almost there. So what did I personally further learn about my teaching experience? So that mainly combination of lectures and practical work work best. Face-to-face, of course, is most suitable for learning skills. And in case online, I already mentioned it, keep the audience active by polls, asking them to activate the camera, breakout groups, all these kind of different things. And that, as mentioned, creating search strategy, conducting the risk of bias assessment, and understanding meta-analysis are so far the most challenging for the participants, but they also find that in the end the most rewarding. This is, I think, the penultimate slide. So the SR process is far too complex for the participants to be able to conduct after workshop. So still then they really need coaching, and not everything sticks. Participants are often not aware that high quality systematic reviews require, besides the topic knowledge, also the involvement of experts. We always say that conducting an SR is a team effort. So you need various types of skills. So skills from a librarian, really the systematic review experts, and also statisticians often. And funding is really needed, especially for coaching abroad. The last thing is the MPMD program, so our political involvement that really worked for us in the Netherlands to get money and to really get our programs going on. So this is also one of the recommendations I would make. So if there are possibilities within Brazil to work via that way, if possible. And that was what I wanted to share about our experience so far. And thank you for your attention.
Thanks a lot, Caroline. We'll now move to the last speaker with questions at the end. But if you have questions already, I mean, you can send them in the chat, either in English or in Portuguese. We'll be happy to translate. So we now move to Alex Bannock-Brown, who is at BAH Quest Center for Improving, I forgot the name, but whatever. She has been in Camaradas for a while, formerly in Edinburgh, now at the Berlin center of Camarade's, and she's also has been involved in systematic review education for quite a while. So up to you, Alex.
Thanks, Olavo. Do you see the presentation in presentation mode?
Yeah, we can see
Yes.
it.
Oh, okay, wonderful, great. Hi, my name is Alex Bannock-Brown. I'm the systematic review methodologist at the Berlin Institute of Health at the Charité University Hospital in Berlin, working at the Camarade's team. My background is in neuroscience and psychology. And like Olavo said, I've been conducting systematic review for a number of years, over 8 years. My role particularly is on teaching review and software and tools for systematic review. And you might also get an email from me if you submit your systematic review protocol at Prospero, because I'm also a Prospero administrator. So Camarade's Berlin is led by Dr. Sarah McCann, and we conduct our own research applying systematic review and meta-analysis, particularly in neurology. Some examples of our current projects include things to do with external validity, so the effect of age and comorbidities, and sex differences in stroke outcomes, as well as a framework for systematic review of in vitro studies. And also on the right-hand side, we provide support to researchers who want to conduct systematic review and meta-analysis in their field. And that includes educational programs, methodological assistance, and access to resources and tools. So it's this right-hand side that I'll have the great opportunity to share with you today. So we have a range of courses designed to increase awareness of systematic review and build methodological capacity and skills. First course is the Introduction to Systematic Review and Meta-Analysis of Animal Studies. This is held regularly, about 4 to 5 times per year. It's open to anybody, provides a general introduction to systematic review of animal studies. We run this 3 days, 3 half days online via Teams, with, again, like Caroline's workshops, both lectures and activities that people can do on their own computer from wherever it is that they're joining. And it's just a general overview and a quick introduction to all of the steps involved. And our course is accredited by the Charité, so we award ECTS points for attending. Of course, our target is we have a general target audience, but this course, the accreditation really supports postgraduate and undergraduate students in their education. We also have a couple of on-demand courses focusing on specific aspects of the systematic review in depth. So our protocol development workshop is 3 full days, very in-depth, where we have small groups working together on every single aspect of the systematic review protocol template. Participants receive tailored feedback from systematic review methodologists, and also evaluate each other's protocols. So they learn to hone a critical eye for both transparent reporting as well as which aspects need to be covered in the protocol. And at the end of the workshop, participants have a protocol ready for submission on Prospero. Our other on-demand workshop is a practical workshop on critical appraisal of primary animal studies. And we run this in a flipped classroom style. So there are theoretical materials, lectures on internal validity and reading that participants must do before they join the workshop. And then the workshop is really hands-on and focused, using flowcharts to work through each of the domains of bias in the circle risk of bias template, to assess bias in a primary animal experiment application that the participants themselves have brought along. So they often bring along a study that's in their field of research, and it really helps them get that in-depth knowledge to assess a study in that much detail. So yeah, really getting into the gritty of study quality and assessing bias. And I also want to just mention some of our e-learning modules and online tutorials. So we have free e-learning. We have about 8 to 9 interactive, self-paced interactive modules where participants can log in and out at their own time. We use the Moodle learning management system for this. where there are both self-paced lectures people can go through, as well as interactive activities, quizzes to test your knowledge, reading, etc. They're open to anybody. All the content is in English, and more information is on the Camarades DE website. We also have a meta-analysis tutorial app. So this is a web-based tutorial using R that walks through step by step the process of conducting a meta-analysis on a published systematic review. No programming knowledge is required, but you can go through it at your own pace. Also, there are quiz questions to check your knowledge. So if people have attended some of our training and workshops and they are excited to learn more, we also offer a number of support services and free resources. So we offer one-to-one advice for research area of preclinical systematic review, and this can range anything from at the very beginning, starting to refine the research question and developing the protocol, to tools and software, data extraction, statistical analysis plan, etc. And we have drop-in sessions, open drop-in sessions, or people can book a specific session with us. We also provide support on how to use SIRF, or the Systematic Review Facility, which is a free software designed specifically for systematic review of animal studies. And lastly, we've also developed a guide, a wiki guide to preclinical systematic review and meta-analysis, which again also has step-by-step instructions, links to resources, etc. for all stages of a systematic review. And 2 years ago, Professor Lino de Oliveira and the team in Florianopolis we worked together to translate this wiki into Portuguese. The wiki is available at camarades.brazil.bio.br. I want to briefly speak about our project titled CAUSE, or Communities for Open Research Synthesis, which is designed to support the widespread awareness of systematic review and meta-analysis in preclinical research. So the benefits of preclinical systematic review, so using data from animal and cells to improve translation, are proven, but systematic reviews are often routinely missing from our research decision-making. So the goal of our project, Communities for Open Research Synthesis, is to bring together researchers and scientists with different expertise to work together to effectively synthesize preclinical research. So our vision is to move forward to a system where preclinical research is appropriately evidence-based, being informed by an unbiased overview of the research data, and that preclinical research being generated contributes seamlessly to the evidence base that we have about a given domain, so that preclinical research is readily synthesizable. So our approach has, we've got 3 approaches. The first is to embed preclinical systematic review education into biomedical training at all German institutions. So to make these methods routinely available, make training routinely accessible, to increase awareness of the methods. The second approach is to scale infrastructure to be able to support preclinical systematic reviews. So actually conducting systematic reviews feasible. And the third approach is to bring together resources and people to grow communities of researchers conducting evidence synthesis to support the community and sharing of best practices and resources. And I just wanted to highlight that the development of this project and these approaches has been facilitated by funding support from the Charité 3Rs department, which has allowed us to establish the range of in-person courses that that I just mentioned, that we've been able to deliver to over 400 people to date. So we're really thankful for that support. So obviously for this, I'll focus on objective 1 or approach 1 about embedding preclinical systematic review into biomedical training. So we're expanding the courses that I mentioned before, offering alternative teaching formats, and I'll share some of our ideas at the end of this talk, essentially ensuring that the education can be taken on demand wherever you are. We're also piloting a train the trainer program with materials to educate additional instructors. And this will enable trainers to educate other researchers at their institutions on systematic review methods in order to effectively deliver education at scale. So these freely available and adaptable resources are intended to build expertise and capacity to carry out systematic review of animal data. And by creating a model that's self-sustaining, we can empower individuals and raise awareness of systematic review more broadly. So building expertise and capacity, second pillar, to scale infrastructure to actually make systematic review feasible. And our third pillar, initiating communities for open research synthesis to support new relationships. So we're expanding our local community, bringing together preclinical researchers in the field, synthesis, information specialists, and statisticians. And this is really a community-led approach. We're creating a digital hub and organizing local and national events to promote networking and the sharing of best practices and resources. Further, we're conducting qualitative research with scientists to better understand personal and institutional barriers and enablers to carrying out systematic review and to educating others. So just to conclude, I want to share some of our ideas for new educational workshops and formats that we're currently working on. They're under development, so stay tuned. The first is an advanced meta-analysis course, and this has actually come from demand from our workshop participants. So things like covering topics such as multiple meta-regression, mixed models, and multi-level meta-analysis models. These models are becoming more and more suitable in order to analyze the quite complex preclinical data. So education is needed in this space. Second, we are designing a learn by doing course. So essentially learn to do a systematic review by actually conducting one, like Alavo and Anna were mentioning. So this is designed as a semester-long course in a flipped classroom style. So the theoretical materials are available beforehand, where small groups of participants work together on all of the steps of the systematic review because they need to conduct a systematic review together, with the end result being a publication which is beneficial for everybody involved. And the third, we are working on a how to make your research synthesizable course. There's large overlap between people interested in conducting systematic reviews of animal research and those who also conduct animal research. So we are designing a course to support people to make their own primary research more synthesizable for systematic review. So discussing topics like primary research protocols, preclinicaltrials.eu, animal study registry, making research data available with metadata, preprint, transparent reporting, etc. So watch this space. And to highlight again some of our key links, all the information is available on our website, cameradi.de. All our workshop materials are freely available on Open Science Framework. And if any of these topics that I've talked about today are of interest, or if you want to implement preclinical systematic review at your institution, we are very happy to discuss, so please get in touch with us. Thank you for listening, and thank you in particular to our funders, Charité 3Rs for kickstarting, Cameradi's Berlin, Volkswagen Stiftung for supporting our communities project, and NC3Ls for supporting the systematic review facility platform.
Thank you, Alex. So we have officially 12 minutes, but I guess we can go on a little longer if everybody can stay for questions. We do have one in the chat already. It's from Pedro Tan, who is in the Netherlands that currently, as I understand. He says, well, I know Brisa efforts are focused on preclinical animal research, but like, what about meta-analysis of translational data from clinical trials, biomarker discovery, bioinformatics, sequencing data? Does that resemble more, I mean, how do we arrive at that? Do we go through the clinical meta-analysis training pathway? Do you go through the meta-analysis training pathway? Do you need to invent something new? And I would actually add, I think in vitro cell line experiments to that. We have some in Brisa, but again, I'm not sure the the usual framework works for many of them as well. So your views on this? I mean, I think the strictly clinical front and the animal front are kind of covered, but like, what about those two different ones? Whoever wants to start.
Yeah, so historically, how the methods for preclinical systematic review developed out of Cochrane, but also required quite a lot of adapting to meet the needs of the data from primary animal experiments. So there's a lot more primary animal studies. The studies are smaller. This obviously needs a different approach, and the same is also warranted for like bench studies, in vitro studies. That's why we're working on developing a methodological framework for systematic reviews of in vitro studies. And I think Pedro also touched on a really important point there about synthesizing data from different streams of evidence. So how do we integrate data from cell studies together with animal studies where relevant? And how do we use, there's a term called triangulation. So using data from multiple evidence streams to help support our decision making. And we can use some of the systematic review or evidence synthesis in general techniques to support us with those decisions. I don't know if any of the other speakers also have anything to add on that point.
Possibly only the fact that indeed the integrating of the various evidence streams is still really a challenge, because those methods for in vitro studies are even further away from being perfect than for animal studies, because it's really a field which starts to grow. Anyway, there are organizations really already busy with developing methods to integrate the evidence, and this is mainly EFSA, the European Food Safety Agency, but also the NIH is working on these integrating evidence streams projects. That was the only thing I wanted to add regarding this.
Anyone else? If you have a question, you can just raise your hand as well, and we'll call you or send in the chat. I do have one as well. And what should be the? I mean, I think from most of your talks, I get the feeling that the main public for this training efforts are actual researchers who do empirical research, who do either animal research in field. I mean, but like researchers who are active at the bench. Do you think that should always be the case? Do you think there's a niche for people who do like only systematic reviews, or for like from clinical meta-analysts who want to try out basic research as well? I mean, who should we be training?
That's really a nice question. Because it's difficult, because the most active ones are and the most enthusiastic ones are the ones who are really planning a systematic review for themselves. But who should we train? I think all basic scientists and all people in the medical field and biomedical scientists already in their bachelor or their master, because you learn much more from these systematic review techniques, and not only animal, about research quality, methodology in general. So who do we need to teach? I think all young scientists, because far too late in their scientific development they learn about these things, but they are more difficult to reach, because in all our master courses they don't conduct research themselves, so they don't really feel I can't find the word. They don't really see the need yet, and they don't really participate as people do who really want to conduct as are on their own.
Yeah, I agree. I mean, we're looking to embed it in all biomedical training programs, at least understanding what a systematic review is, how to use one in designing an experiment. I think that's really important for the 3 Rs. Obviously, where systematic reviews are at the moment is that not every single topic already has a high quality systematic review out there. So where people often see there is a gap, as in they have a research question, but it hasn't been synthesized, the evidence hasn't been synthesized to date, then often people go in and think, okay, then I can do this. Want to learn more, but I think everyone should learn how to use the systematic review in their research decision making.
Maybe add to that a little bit more. I would like to say that I think that so for me, it's not even that important that you really learn, yeah, of course, as well, learn about systematic review technique, but many of those steps about really a good search, conducting a really comprehensive search, being aware of quality could be individual steps and not that much related to a systematic review. But I think that the systematic review on itself is a very nice way to teach people about many basic concepts of research in more detail.
Yeah, that's what I would add. I thought it was really interesting. I didn't knew about those training of specific issues that Alex talked about. And for example, specifically training on search or in quality of studies is something that could help to any researcher. Actually, I have already had the feedback while conducting systematic reviews with researchers that haven't done this before, that how they started thinking how this could change their work as basic researchers, like what they could change for them, their work to be better than those that were being evaluated during the systematic reviews.
So we got another question from Anna Ehrman. So she mentions that, well, everybody mentioned risk of bias assessment, but what about dealing with possibly fraudulent studies? And what should be the approach with studies that's sufficient, not plausible, based on results only? And I think that's actually like a very pressing question. The clinical field, Cochrane has changed their methods. I think maybe Thais can comment. And I do think that problem also exists we've done cell line studies. There's like completely implausible results. I mean, this just can be right. You have like an effect of 100, but I mean, what do you do? I mean, that's to me, it's a pressing question on what we're doing right now.
Yeah, I mean, sorry.
Okay, go ahead.
No, Thais, go ahead.
No, I would ask you to repeat your question.
How do you deal with possible with studies that are possibly fraudulent or that just do not seem plausible because of the data or the results or whatever? I mean, should you just
the systematic review is not feasible, you mean? Or
no,
the originals, the basic study?
The original studies. I mean, original studies that you think that should not be believed because of X, whatever. I mean, they will fall out there. I mean, if
you believe it's a fraud or something like that?
Yeah, or
well, in your eligibility criteria, you can provide which designs that are eligible, and then also in your risk of bias assessment, you can like put threshold or something if the area is has problems like this with fraud. Usually in our epidemiological systematic reviews, we do not exclude the studies based on their risk of bias, but we do judge this and would do sensibility analysis, checking if this result impacts in the new evidence, and so we discuss if it would be better to provide the result without this and then discuss. But then it's something that your method, mainly because you're aware of problems in the area, then you anticipate these problems in your method. That's why piloting and checking the studies prior to initiating your systematic review would help anticipating this. But sometimes the challenges would happen meanwhile you were performing the research, and that's why have a good supervisor, good coaching in these activities would be really helpful.
And to add to that, checking which studies have potentially been subsequently retracted by the journal. I know that this is a topic that involves multiple like actors as well as like reviewers, publishers as well. Publishers are also becoming more aware of this and investing into tools and software to support picking up fraudulent activities. I don't know if anyone follows Elizabeth Bick on Twitter. She calls herself a scientific sleuth, and her perception is amazing. She can detect fraudulent or adapted images and Western blots and things like that, where people have actually copied images. So yeah, it's a topic that doesn't just affect us in systematic review, but as Thijs said, adding these methods into your protocol, or like Gabriel wrote in the chat, performing a sensitivity analysis to exclude the studies that you believe could be fraudulent, that would also be able to help there.
To add to that, it remains challenging anyway. We have a large systematic review now in our own department, on which we expect that a large part, and we never had that before, is fraudulent. But we can't really make it clear. So we do multiple things. So we do indeed conduct a sensitivity analysis. We also check whether or not the authors, the initial authors, are affiliated to really a university. Often they have these emails with gmail.com and are not affiliated to universities. But essentially, my main thing is that if it's really a good fraudulent study, we just don't know. And it's very difficult if you say, okay, we think this one is fraudulent, why the other paper from the Netherlands or Belgium isn't, or Russia or China. So it's really a problem. And I think we can do our best, and our sensitivity analysis help, but I don't think there's really a good method now. There are efforts, but as a reviewer myself, I often just don't have really a clue about how to deal with these studies, and to be sure.
I agree. Good fraud is there, but there's a bad fraud. This obviously looks fishy. What do you do with it? But still, there's a bad fraud that you can reprove consistently, and I think that's really the threshold. Because I can do my human, my personal decision, just like not taking this into account by myself, but like I cannot do it in a systematic review. So like, how do you make those decisions explicit and unbiased? Is actually very hard. More questions? I have a couple more. Gabriel.
Hello. So thank you very much for all your lectures. That was fantastic. And I have something like a comment. I am working with a research group in the state of São Paulo, trying to translate the circle risk of bias tool into Brazilian Portuguese. And I guess I mentioned that to Caroline several months ago. And we did that with back translation and cultural adaptation, and the tool looked beautifully. But then we tried that and tested if people could implement that. And people cannot implement that without training. At first, we tried with a group of students, asking them to rate, based on the Brazilian version of this tool, to rate a group of articles. And the agreement rate was terrible. And we thought, hey, these are students, so they might have no experience to do that. Then we tried again with professors, and it was even worse. So the comment is more like to reinforce the importance of training. Regardless of how good the tools are, they are not necessarily self-explanatory. So that's the reason why you should spread the word about the relevance and the importance of getting people duly trained. Otherwise, things will not work.
Yeah, I totally agree with you, Gabriel, because this is exactly what we experienced. How much signaling questions you phrase, how much explicit you want to try to make those tools, people interpret it their way anyway. So the only thing, what we typically do is, for each systematic review, the team I supervise, for example, so I'm not talking about the training, but really about the supervising during the conduct, we let them in their entire team score the same 10 papers, and then we discuss all the discrepancies with me by phone, and I do the same 10 for them as well, and then we discuss. And that's the only then they really start to grasp, I think that's the word, the essential and the difficulties in rating risk of bias. So indeed, without training, I don't know how it should go right.
That's like one of other exercises in our workshops is we have people work on it together, and then they need to collectively present what the reconciled decision is. And that really, it like the switch, it's a light bulb moment. Oh, it's so subjective. And that you actually need to have a really deep understanding of the primary studies. I think this is why it's really, really important that just a team of statisticians couldn't conduct a systematic review on a topic, because you really do have to have the topic knowledge, the domain knowledge of where all the different types of bias can creep in to this type of experimental work. And yeah, it's really important that the training and working together to actually get the reconciled decision for these bias items.
Yeah, and I guess the comment touches on evaluation and how do you evaluate your efforts. I think Carlin touched on this via qualitative studies with people who went through training and their own views, which I think is a great way to do it. But how else can you do it? I mean, if the others can comment, do you have any evaluation of how of what your training actually achieves? And I could think of other methods. Like, can you actually see how many of those people who went to the course actually completed a systematic review at some point? Or maybe even rate those reviews. I don't know, like what other approaches to training you see, and what have you tried in your own courses?
Yeah, so we have it's actually a 2-year evaluation follow-up for everybody that participates in our workshops, based on the Kirkpatrick model. So in the initial straight after the workshop or soon after the workshop, they receive information about kind of initial satisfaction, and then 6 months, and then 1 year after, about how much these the things that have been learned in the workshop are actually implemented into the daily work. I didn't present any slides about that today, but I can send you through some more information on how we follow up people over years to see whether this training they've received actually has an impact on their daily work.
Yeah, and in our case, the people who attend our workshop generally, those are the ZonMU workshops. They only attend the course because they want to conduct their systematic reviews. And if they only get the money to conduct this SR if they are supervised by either me or Kim, my colleague. So in our case, these systematic reviews are not formally evaluated for their quality, but I don't want to be. So if I'm an author, I agreed and generally with the methods they used. So that's how we do it nowadays. In that way, we make sure that the quality of the systematic reviews after conducting the workshops is how we want it. But it's very interesting, as you say, Olava, to really evaluate all those systematic reviews, and especially the ones after, in our case, the project, the program, how they will do without supervision afterwards. This is a very nice, interesting project.
How well do the tools adapt, though? Because I mean, we do have like MSTAR and other approaches to evaluate meta-analysis on a clinical front, but do we actually have a way to, like, Prisma was not meant for this, it's a reporting guideline, right? So how do you rate the preclinical meta-analysis? Do we have a tool for that?
So the Prisma for preclinical studies is under development. I think, Carla, you're involved in that as well.
Yeah,
obviously it's reporting.
But it's
just a reporting guideline.
We don't have MSTAR pre or something, so yeah.
We don't have MSTAR pre, but there's also the ROBIS tool from the Bristol group for evaluating the quality of systematic reviews generally, I think. Caroline, maybe you can speak more to what you use specifically when you evaluate the systematic reviews that come through the ZonM.
Yeah, but we didn't evaluate the actual, as I mentioned, not actual systematic reviews themselves. We evaluated the process, and the systematic reviews, they are agreed by us, but not evaluated by us. So if we don't agree with the methods, then we already intervened along the way. So at the manuscript phase, we can't evaluate anymore. But I think indeed what Alex is saying, that Robis is the tool to be used here. And I think that we can use it, also adapt it on some minor parts, but generally the same things hold for preclinical SRs as well, only about when you're talking about external validity and translating and indirectness. We have multiple levels, of course, of indirectness for animal studies. There are some adjustments, but I think for 90%, the Robis tool is also sufficient for preclinical SRs.
Yes, beside all Robis, there is AMSTAR. AMSTAR tool is for assessing the quality of systematic reviews, but also will check the process and not the certainty of evidence, but if the process is transparent and well performed.
So, more questions? Well, if we can go with the final.
Oh, sorry, Alava. I actually have a question to Mariana about your community on the platform. I believe it was, is it Zulip? Is that correct? How did you find, I mean, it was really interesting to hear how you kind of had an application process for people to be involved in Brisa, and then the subsequent kind of community conversations on this online portal. Do you find that this communication is very, very project specific, or do you see other sharing of resources and connections that are that continue to be maintained despite a Brisa admin intervention?
Well, actually, in the beginning was kind of difficult for people to get into Zulip, and we have like a more or less engagement. So it's something that we are using and it's being effective, but we still have problems for the use for the projects yet. So I would say that it could be used for more than the discussion on the project itself, but not everybody in Brisa is really involved. So a little bit of insistence and getting used to it is still needed.
Yeah, I mean, we try to enforce it as Brisa coordination, because I mean, it helps. I mean, if everybody talks about the projects in an open space, we kind of like know what's happening, and we can also contribute to. I mean, people can contribute to other projects. I mean, we can just like call on everybody. Like, well, it's much better than having. If you haven't used Zulip, it's basically an alternative Slack, an open source. But I think like channeled communication works really well to manage those larger collaborations that work on like many specific projects, especially for those supervising, which is in the case Mariana, me, and Anna. And but again, I think it's not natural for most people. Most people will try to have to do their own WhatsApp group, which is I think bad for the whole group to know.
Transparency.
But we still depend very much on monthly meetings as well. So we do have a general Brisa meeting every month in which people present they give updates on how the project is going. I think everybody talks. So I think that's the mainstay. We try to work offline on Zulip as well. Some groups do that really well, and I think can coordinate around that. Some are more, are more resistant to the platform. But
cool. Did you do like an introduction to Zulip with the participants? So, or like an onboarding? I'm wondering, I mean, some people take very intuitively to a new software, but others, it takes a while, which I think everybody experienced during the pandemic. Someone invited you to a call on yet another platform that you'd never used before. It took a while to get used to. Was there some sort of onboarding or training for this?
Yeah, actually we haven't, but it could be a good idea, maybe.
I mean, we talked about it in the general meetings, right? I mean, informally we did, but I mean, we never did have like a formal onboarding. Maybe Ana Paula can take note of that.
We did some help for people to get into it, and Ana Paula actually did it, but not an onboarding on the specific tools. And maybe it could be a good idea. Maybe it could help in that challenge for more people to use it frequently.
Cool. Yeah, I'll definitely get in contact with you. I have many more questions about this platform. Thanks.
Any more questions? Well, if I can do a final one, I think this is like the final one in a lot of events these days. Like, what do you see the future like in terms of like technology getting making us obsolete? I mean, Alex has done some work on machine learning but in very specific applications during search. But I mean, I don't think we're very far from the point where like a language model will just like take over, read all the articles, and output something in, I don't know, 5, 10, 15 years at most. So like how do you, I mean, what should we be doing to prepare for that in terms of training people to actually like work with these technology tools? Because I think again, data extraction, I mean, not only screening but like selection, extraction, etc., will certainly be outsourced technology at some point in the not that distant future. So what should be, I mean, what steps should we be taking to just prepare for that and prepare people who we're training for that pretty near future, I'll say?
Yeah, I love this question, especially since ChatGPT came out. I think Caroline and I have also talked about this quite a lot. We get this question every time we do a training. I would love it if there was a machine that you could press the button and you could put in your research question and it would do a systematic review for you on the spot. I think we're quite far away from that at the moment. One of the main hurdles is that scientific research is published in a non-machine readable format, still PDFs. Data is provided as a bar chart with error bars that are not specified. So we still have to extract, I mean, machine assisted extracting the quantitative summary data from graphs, which is why we also want to focus on how you can make your research synthesizable so that it's easier to get this data out of public studies. The thing, I think this has large implications for training. So we already talked a lot about risk of bias assessment. You also have to have in-depth understanding of each of the domain knowledge, and 2, the methods of the systematic review to check the output of the automation. You will always need to check the output of any automated tool. For example, if you ask ChatGPT for give me 10 references about systematic review of preclinical data, 2 or 3 of them are completely made up references. It will say, oh, a paper published by Mariana and Alex and Olavo and Caroline and Thijs was published in 2021 entitled Preclinical Systematic Review. Unfortunately, we never published something together in 2021. Maybe we should. But it just completely makes it up. It understands what a reference should look like, and it just pastes in people that it knows are roughly in this domain. I understand that the rate of how these models improves at the moment is exponential, but it learns based on published data. So the reporting quality of published data is not good, which we know from having conducted systematic reviews. It's impossible to know everything that was conducted in an experiment based on the published report. So until reporting of experiments is fully transparent, we will not be able to do this because we still make inferences. Oh, did they say they did blinding? Did they do it for every outcome? Not sure, question mark, unclear. If we don't know, the machine doesn't either.
No, I agree, but I just don't think we're that far from actually having something that can spit out a very convincing systematic review. It might not be a good one, which is why I'm worried about this. But I mean, just to mention, I mean, you have Neurosynth, and I'm not sure if you're familiar with it. It's been around since 2011. It's the work of Tau Yarkoni and Wes Podrek, but it's actually a very primary attempt to do automatic meta-analysis in fMRI, which is a terribly complicated methodological field, but it's I mean, it worked based on the fact that like most table ones look the same, so it's about like data standardization. But I mean, it pretty much read through the article, found words that were over a given frequency and associated that. I mean, it was very primary, but with models that actually understand text, which we have now, I think you can go like way over that. And actually extracting data from graphs shouldn't be that hard as well. So I mean, it looks pretty feasible, but again, I agree with you. There's like a lot of tacit inference going on, and I don't know, yeah, but again, it's something that will spit out something in a minute versus 3 months of work. So like people will be doing it. So I think we should be preparing for that.
I think that to add to that, we also, it's important that we keep evaluating the tools that are out there. In a large project with EFSA, we're currently doing that for multiple tools on different levels for systematic review. So you were asking Olava about the step, what I think that we need to take. I think that that's one of the steps. So keep on track on what is out there and what's useful. And for example, I recently did that with our consortium in which Alex is involved as well, for tools available to create comprehensive search strategies, and a conclusion of all the tools out there. There's nothing which works sufficient. So in the end, you need to do it on your own. And this should be relatively easy. And if we're talking about, for example, as Alex touched upon as well, on risk of bias, that's even more challenging as it's a judgment. So for example, when we're talking about blinding, you're not looking for the word blinding, you check if blinding could have been broken. So maybe that one animal had an extra tube which the other didn't have. So that will be so challenging to automate. So I hope, and I'm on the same page as Alex, that I hope that we can do it in 15 years, but I'm doubting. But in the meantime, evaluate those tools and integrate them in the courses, because in each course we give nowadays, people ask about tools. Can I use ChatGPT? Can I use this? Are you aware of that? So yeah, we need just evaluate and tell them the results so far. And especially also explain the risks of using those automatic tools which are out there right now.
Anyone else? Well, I think this makes it then. We're 20 minutes late already. So, well, thanks. Gabriel, oh no, that's applause. Okay, sorry. So, well, thanks very much, guys, for all the amazing participations. I guess we've learned a lot. I've been thinking a lot about what has been said here. We should getting in touch with some of you guys concerning some of the stuff you're doing, because it might interest us. Alex, particularly the risk of bias tools for in vitro studies, something we've been thinking about as well. So maybe we would like to know. I mean, people from Brisa might be interested in contributing. We have some interested members in that area, so it would be nice to exchange some ideas afterwards. And thanks very much for your result. I mean, everybody I think mentioned open resources, which I think is great as well. And I think perhaps most of the people here are actually more interested in knowing where to learn than actually engaging in this training discussion. But so, I mean, thank you very much for what you put out there. And I guess we can serve as inspiration for each other. And that's it for me. Thank you. And hopefully it's been interesting for you as well.
Thank you all. Congratulations for the initiative. And anything you need, I am also available.
Thank you so much for the invitation. Yeah, hopefully speak soon.
Yeah, and again, if I can be advertising as well, I mean, if anybody's interested in reproducibility in a wider scale, we can also meet at the launch of the Brazilian Reproducibility Network, that time in Portuguese, next week, June 28th at Wednesday at noon. So, and the Brisa material, of course, is online. I'll put the links again here, just in case somebody got here late, but you can all access our course material free until 2024, when we plan to have a new call, and hopefully have more Brisa members coming in.
Bye.
Bye.
Bye.